On the next slide, perfect. We'll also do a bit of beekeeping Q&A at the end. And of course, Joe will do our door prizes in person now. We're very excited. On the next slide, I want to share with you the upcoming Texas Beekeepers Association convention. So if you guys aren't familiar, every year they throw a really wonderful convention for us here in Texas. It's not always in Galveston, so this is kind of a treat to have it close. But they have a 2-day kind of agenda-packed conference, all kinds of speakers from across the nation. And of course, you'll see a lot of us there, as well as a lot of your fellow beekeepers from around Houston and all over Texas. Everybody kind of comes down and spends the weekend at the beach. So if you haven't thought about it, do think about joining us. It'll be a lot of fun, some great courses. They also will have a whole area where you can do all your shopping. So if you are like me, I tend to collect all kinds of bee goodies. And so they'll have plenty of those for us too on the 2 days. So with that, what's next on our next slide? Just in case I've missed anything. Oh, I haven't missed anything yet. So I am really excited to share with you Jordan Ellis, not to be confused with Jamie Ellis, but Jordan Ellis is here with us today. She is a graduate of Cornell with her degree in insect biology. And now she is at A&M doing her graduate work for a PhD. And today she's going to actually talk about the social immunity of honeybees. She spoke at the last beekeeping conference or beekeeping courses that were up in the Bryan College Station area just a few weekends ago. And she's really got some amazing insights on how bees really support each other in their own health, and then some great research that she's been doing to share with us today. So with that, I'm going to turn it over to Jordan.
Awesome. Hi, everybody. Is my audio good? Are we good? Okay. Just figure out my screen. Nice. So should I share my screen? Here we go. Okay. All right. Is this good? Okay. Hi, everybody. Thank you for that introduction. I'm happy to be here. Happy to see all of you. So I am going to be talking about social immunity in honeybees. So yeah, let's get started, I guess. Okay. So first up, I want to bring up a classic honeybee example. And this here on the side of the screen is the murder hornet. So, and then over here where the honeybees are, these are the Asian honeybees, Apis cerana. And so what we have here is a predatory wasp that will try to come into these honeybee colonies and take over. They'll try to eat the honeybees and then essentially take over the honeybee colony so that they can parasitize the brood and consume what's in the colony and drive these honeybees out. And so this is a big threat for honeybees, right? Especially the Asian honeybee. And so in order to deal with this threat, what the honeybees do is they heat ball these murder hornets. So I think this is just one of the coolest things about honeybees, because if you put one honeybee up against one murder hornet, there's no chance, right? They don't stand a chance. This hornet is going to eat them and there will be absolutely nothing left of them in the end. But what happens here when you have a whole group of honeybees is that they're able to overwhelm this hornet, and they will all gather around it and heat up their wing muscles to a temperature that's above the survivor temperature of the hornet, but where the bees can still survive. And so they kill the hornet by essentially just like broiling it alive in a lump of honeybees, which is so cool. And so this is an example of social immunity, because you have this interaction where these honeybees are combining and using their social structure to get rid of a predator in this case, but it could be kind of anything in social immunity. So they're combining their resources to get rid of some invasion in their colony. So this is my favorite example of social immunity, because it's just really wild. And it's one of those things that you can tell people what's the coolest thing about honeybees. And you can tell them that they heat up hornets until they die. So this is kind of one of the things that we're going to be talking about today, and kind of how honeybees exhibit other forms of social immunity that are maybe less obvious to us and a little bit less flashy. So we can move on here, and I'm going to talk a little bit about what social immunity is in general. So social immunity is the level of disease protection that is conferred by the social group, like the collective group. And so it can be any sort of defense against diseases, against parasites, against predators. And so it's something that, you know, the bees wouldn't be able to defend against this hornet all alone, but when they're a social group, they can. So also just a little bit more background is, of course, eusocial insect fitness. And this is why honeybees are interested in defending their colony as a whole, right? So with honeybees, they are more related to their sisters than they would be to their own progeny. And this is because of their sex selection ratio here. So they're 75% related to their super sisters, which means that they're more interested in their colony survival than they are in their own individual survival or in their survival of their progeny. So this means that their fitness is best served by acting in the interest of a colony as a whole. And so again, this means that they can kind of team up to take on threats to the colony. And they would prefer to team up to take on these threats as opposed to die, because their fitness is best served when the colony continues to survive. So again, that's just reiterating that colony survival is more important than individual survival in this case. And a great example of this is stinging, because we all know that when a honeybee stings, it does die. And so this is kind of an example of this altruistic behavior, wherein the honeybee is trying to get their colony to survive, sometimes even at the sacrifice of their own fitness or survivorship. So this brings me again back to social immunity and what it kind of can defend against within the colony. So we have social immunity as a defense against predators like these murder hornets. And then we're also having social immunity as a defense against viruses. One that obviously everyone is pretty concerned about is deformed wing virus. And so we're going to talk about how social immunity can defend against deformed wing virus as well as other viruses, but this is just a really prevalent one. And also we're going to talk about social immunity defense against fungal infections such as chalk brood. And then lastly, I did just want to bring up the Varroa Mite just for a little bit of background on the Varroa Mite, because this is obviously one of the most important parasites and important things in honeybee survivorship today. So the life cycle of the Varroa Mite, I have this figure over on the side of the screen here, and this just shows the life cycle of the Varroa Mite in conjunction with the life cycle of the honeybee. So we have Varroa mites that will infect the brood right before it's going to be capped for pupation. So a Varroa mite will go into the cell and then it will parasitize the honeybee as it pupates into an adult bee. And so it'll feed on the honeybee, and also it will be a female mite, and she will reproduce within the cell. She'll have one son mite and multiple daughter mites, and they'll interbreed. And so then when the honeybee emerges as an adult, there will be multiple fertile mites emerging with her that can go on and proliferate and, infect the rest of the colony. So we're going to also talk about how social immunity can sometimes defend against this, and how, we can see examples of this also in other sorts of honeybees. And then another parasite is tracheal mites, and so we'll touch on that a little bit as well. And so I just wanted to kind of give a background of all the things that social immunity can be good for, because it's kind of the whole gamut of what we're working with in honeybee colonies, because it's this social thing. So anything that's kind of evolved within honeybee colonies, these honeybees have sort of an innate defense against. So that's kind of what we're going to be talking about, which brings me to all of the different mechanisms of social immunity that I'm going to talk about today. So this is quite a big graphic. We're going to go into each one of these different mechanisms of social immunity, and we can, we'll get into it a little bit later, but all of these mechanisms come from this paper, which is a review of social immunity, and it's pretty interesting. So I've got to give credit where credit is due in that case. And so what we're going to be talking about here is different forms of social immunity that range from constitutive mechanisms. So this means that they're in place within the colony all the time, right? So one of the examples of this is polyandry. So this is how many drones that a queen mates with. And this is something that's going to be constant in pretty much every honeybee colony. This isn't something that the colony as a whole has control over. So this is something that we're calling constitutive, and it's just kind of in place within the colony. And then we'll also range over to what we're calling inducible traits. And these are things such as with this murder hornet that can be kind of turned on a dime. So when the murder hornet comes to infest the colony, all the bees will come and swarm the murder hornet. And so that's an inducible form of social immunity. And so there's all these different forms of it. So I guess I'm going to go over each one individually, and then I'll talk a little bit about how this can apply to everyday beekeeping, and what implications it has on maybe your bees. So to start out with, I'm going to start with polyandry because this is the most constitutive one, and it's the one that will just kind of be in place within the colony. And how this applies to social immunity is that queens will mate with 10 to 20 drones on the whole, and this allows her offspring to have more genetic diversity. So the more drones she mates with, the more differing genes her progeny will have. And it's been shown in multiple studies that this can actually confer levels of disease resistance. So this image here shows if a queen mated with a bunch of different drones, all the different potential genotypes of their offspring, and you'll see that they're super variable. So there's all these different colors going on. And so you can imagine that if one of these colors of genotypes has, some sort of mutation where it's not as good at nursing, or it's not as good at foraging, or it has something wrong with it essentially, the fact that she mates with more drones will help cover that up within the colony, and these bees will be better at performing tasks on the whole because they have the range of different genotypes, so they can kind of correct for each other's problems essentially. And so studies have shown that colonies with higher genetic diversity forage better, they have healthier gut microbiomes, and they're more resistant to diseases such as deformed wing virus. So this is really cool, but obviously within your apiary, it's not really that applicable because you can't really control how many queens or drones your queen mates with. So I did try to put a takeaway for each one of these things, but obviously this one isn't really something you can control for. So the queens will naturally seek to mate well, but if you're buying or breeding queens, you can make sure they're naturally mated in heavily populated areas. So there's just a lot more drones. But obviously this one's kind of just luck of the draw, fingers crossed that you get a good queen. So then we're going to move on to some more different constitutive mechanisms. And I think the next one up is task allocation. So this one is one of the ones that I find the most interesting. And this has to do with age polyethyism within honeybees. And so I'm sure you've heard about this before, but this is essentially the division of labor that bees perform within their life cycle. So after a bee emerges from pupation, she'll perform different jobs as she ages. The first one we see over here, right as she's emerged at the top there, she'll start to clean out her honeycomb, like where she emerged from, and then she'll start to clean out the rest of the comb, be a little cleaner bee. And then as she ages, she'll take on nursing roles and she'll nurse the younger larvae. And then as she ages even more, she'll start to help build honeycomb. And around 2 weeks, she'll start receiving food from incoming foragers and moving towards the outside of the nest, where until finally around 3 weeks of age, she'll become a foraging bee. And so this is not only just really interesting that this happens, but it also does serve kind of a disease protection purpose. And this is because the younger bees are performing the tasks within the colony. So as a bee emerges from pupation, presumably she's not very sick, she's not infected with any diseases. And then she starts to, around 1 week old, she'll start to feed other larvae. And so because she's a bee that hasn't had any contact with the outside world or with, other flowers or anywhere that has heavy disease prevalence, she's not going to have a lot of disease in her when she feeds these young bees. So then that keeps this cycle of like not having a lot of diseases within the bees. But as the bees age, they move outside of the nest, of the brood nest. And so foraging bees will hang out kind of around the brood nest instead of in the brood nest so that they don't take their contamination essentially into the younger bees, and they keep the colony more healthy. So this one I think is really cool just because this is just the natural progression, and it functions really well within the hive, but it also, I think I have another slide on it. Yeah, this is just a little diagram where you have the nursing bees that are in contact with the larvae, and these are going to be the healthy bees. And then once they go outside, that's a more dangerous role, more pathogen-rich role. They're going to be in contact with deformed wing virus, potentially more Varroa mites. So when they're outside, they'll come back within the colony and they'll stay to the outside of the nest so they aren't in contact with the future of the colony, right, with the younger bees. So they keep the future of the colony as healthy as possible. So I find this one really cool because this is just, kind of a natural mechanism that happens. And I also, a lot of my research has to do with age polyethyism. So I think this one's really interesting. And a point here is that even like middle-aged bees, if they're performing undertaker roles where they're in contact with dead bees that potentially have high viral levels, they will also hang out around the outside of the nest so they don't contact the young bees. So in terms of how this one can apply to your bees, obviously all of these things are kind of how the bees protect each other. So there's not a ton that you can do with this either, but I put some things in here just for fun. So I thought that, you know, leaving brood in the colony is good, not messing with the brood ratios, not messing with what the colony itself is producing, because I know in our apiary sometimes we'll interchange brood with colonies that need a lot more brood or are sick or unhealthy, but then that can kind of inconvenience the colony that you're taking from. So just maintaining those ratios is really important, because if you cause a gap in that age range, then you're going to have bees that are trying to take on those jobs that aren't the right ages, and then you could get this cross-contamination. So yeah, that's that specific aspect of social immunity. So now we're going to move on to this one, which has a really fun name, and I'm just going to change it because I've called this the use of self-produced compounds. So this is another kind of constitutive mechanism where all bees are self-producing compounds, and they all have sort of mechanisms that are providing a protection for the colony. And so the first one up is venom. Like we talked about earlier, bees can sting, and this is kind of an altruistic form of protection of the colony. But venom also serves another purpose, which I didn't know until I read this paper that talks about it, is that honeybee venom has antimicrobial properties. So not only is it like a protective mechanism against predators, but it also can protect against diseases too. And so it can be found on the honeybee cuticle. So they'll kind of have venom on their cuticle, so as to protect against diseases that are like entering their trachea or anything like that. So venom can actually be an antimicrobial protection agent, which I think is pretty cool. And then another use of self-produced compounds is brood food. And so this one is that there's evidence that colonies invest in production of larval food that's higher in antimicrobial peptides. So again, that when they're feeding their younger bees, they're providing these antimicrobial properties for the young bees so that they can defend against any sort of pathogens that they come in contact with, or so this also, again, we're trying to keep the young healthy so that they can age up and be a productive colony as they get older. So this just protects the future of the colony. So I think there's one more. And so this one, obviously, prophylaxis. And this one's cool because I didn't know that this was a social immune tactic, but it actually turns out that engaging in prophylaxis does influence disease susceptibility within the colony. And this can be because bees that have, low-level infections will perform prophylaxis with bees, and it will like prime their immune system so that when they come in contact with these pathogens within the real world, they'll have an immune response ready to take on those pathogens. So when bees, within the brood nest perform prophylaxis with each other, it will confer this kind of immune response that allows the bees to take on more pathogens when they actually come in contact with them. So this could be an element in colony level pathogen defense as well. So we have all of these things that are, produced by the colony, a product of the social structure itself, that are again helping cause antimicrobial or antibacterial components within the colony. So it's protecting against different pathogens. And so for my takeaway for this one, I thought was a little more solid because what I said before is that the brood food is made from certain pollen, and the certain pollen has antimicrobial activities. So for this one, I said that allowing bees access to many types of pollen can help them access the pollen that does have antimicrobial properties. And so obviously this kind of just goes with allowing bees to have access to a large range of pollination sources. But I think this is cool because it kind of provides a background for why you would want to do that. And it's because they have antimicrobial properties and can keep the colony safe as a whole. And so then I also said, if a colony has a lot of excess pollen, you could freeze a frame that you can later use to bolster colonies that are weaker to allow the brood to continue to have this antimicrobial properties to defend against the future pathogens that they encounter. So moving on more, we have resin use, and this is in the form of propolis. And this might be one that people are familiar with, but propolis is essentially a plant-produced compound that honeybees will collect. It's resin, and then they kind of mix it with honeybee compounds, and then they will stick it around their hive to, decrease certain areas, make the entrance smaller, coat the inside of tree cavities if they're nesting within a tree cavity. And, in your honeybee colonies, they're always in the parts that you're trying to open. And so this is, kind of annoying, but it also turns out that propolis has a lot of antimicrobial properties and can be a direct defense against some pathogens such as chalkbrood. So that's the fungal pathogen that we're working with. And then it was even observed in one paper that a subspecies of Apis mellifera uses propolis as hive beetle traps, so they get stuck in them and they can't continue to parasitize the colony. So this one's really cool because propolis is really interesting. And this is one that, we as beekeepers are I want to scrape the propolis off whenever I open the top of the colony, but it's actually helpful for the colony as a whole because it does have these antimicrobial properties. And so then again, on this use of plant-based compounds, we have honey, and it's no shocker here. Honey also has antimicrobial properties. We're learning that a lot of the things that bees produce have antimicrobial properties, which again is really cool because they're kind of doing this themselves, and it's this innate disease response that they have within their colony already. And honey is another one. So sick bees sometimes will actually self-medicate with honey that does have higher antimicrobial properties so that they can kind of try to drive out the pathogens within their system and medicate themselves, which is really cool. And so for this one, again, for takeaways, bees do it best. So when it's not super bothersome, you can leave the propolis within a colony, especially when it's kind of at the entrance, because that provides a little antimicrobial gate when the bees walk in to keep the colony safe. And then also because honey from certain nectar has antimicrobial activities, continuing to allow access to different floral resources for nectar is good. And then also trying to leave a little bit of honey once you extract honey can give them a little bit of a boost overwintering. If they come in contact with any pathogens over winter, it'll allow them to self-medicate and keep driving those pathogens out, which is cool. Yeah. So then moving on more, we have this aloe grooming as another social immune responses. And you can see here, we're moving more towards the inducible traits. So these are ones that can kind of happen whenever they decide to cause them to happen, instead of it's just always happening within the colony. And so grooming is a good one because here we start to get into the range of the Varroa mites, which we always want to be defending against. So honeybees do participate in grooming each other. And this is helpful against both the tracheal mites and the Varroa mites because it can dislodge and then also injure the mites. So we see a picture up here in the top, that's a Varroa mite that has all of its legs. And then in the bottom here, we see the top kind of 2 legs, and the ones on the side have been nibbled off by a honeybee that's trying to protect the colony. So yeah, so this one's good. And then similar to with the prophylaxis, because grooming is performed, with mandibles and such, it's thought that grooming of diseased individuals by healthy individuals also provides this low level immune priming response so that the bees get this small level infection so that their immune system is ready when they come in contact with these pathogens on a larger scale. So yeah, this is another really cool one. And my takeaway for obviously you can't cause grooming within a colony, and there's really not a threshold. I looked into this a little bit because I wanted to have a takeaway here, but there's no assay that you can do to kind of evaluate how good a colony is at grooming. And I thought that was kind of interesting because if we could do that, and I found one where people were trying to use flour to coat the bees with flour and then observe how much they groomed within a certain period of time. Because if we could do that and observe how good they are at grooming, then you could potentially create a line of bees that's even better at grooming, and try to keep using these social immune tactics for the benefit of the bees. But yeah, also we have our powdered sugar bees after a sugar shake because they're really cute. So moving even more towards the inducible behavior, we do have hygienic behavior. And this one you might have heard about because this is kind of a big thing these days. Hygienic behavior is what happens when you have brood that's infected or dead, particularly by Varroa mites, and the bees within the colony will detect and remove the infected brood within the colony. And the pupa that's removed will have the parasites and or pathogens in it, and then they'll try to discard that outside of the colony so as to protect the colony from these parasites and pathogens that they're removing. And so this is really cool, and this is a behavior that we do see within colonies, and we think it's conferred by the pheromones that are given off by the brood. But this is one of the ones that can vary heavily with colony to colony, how much hygienic behavior they're performing. And similar to grooming, this is actually one that we do have assays to see how good a colony is at hygienic behavior. And this is an assay that is done with liquid nitrogen. So you'll take a frame of brood, put basically a PVC pipe right in the middle and pour liquid nitrogen on it to kill all of the brood in that area. And then you can insert it back within the colony and leave it for 24 hours. And then when you remove it, you can look to see how much brood is removed from that killed area, because this killed area of brood, right, won't be giving off a good brood pheromone. So the honeybees within the colony should be able to sense that this is dead brood. And if they're good at performing hygienic behavior, they'll remove all of the pupae from the comb, like this one here on the left side. And then if they're not good at it, you can see on the right side, you have a bunch of pupae that are still in the circular area that are dead. And so this colony would be not as good at performing the hygienic behavior. And this is important because all those bees within those capped cells in a real colony could have Varroa mites within them. And so if the bees are not able to acknowledge the cells that have Varroa mites and get rid of them, the Varroa mites are able to proliferate even more. So yeah, so the rate of removal is correlated with colony level resistance to bacterial and fungal diseases, as well as Varroa mites. And so this is one that, because we have an assay to kind of look at how good it is, how good a colony is at hygienic behavior, we are able to breed hygienic lines. But hygienic lines, I think, are pretty expensive. So it's not really something that you can just go out and buy. But it is kind of cool because it does show that we can work with these social immune techniques to try to help the honeybees help themselves, essentially, and maybe help them out in a way that's not intrusive to their behavior, but maybe encouraging certain parts of their behavior that are helpful against certain things like this. And so I do have an example of hygienic behavior being important within honeybee colonies, but this one's kind of in deep, so I'll try to explain it really well. But you can ask me any questions if you're struggling to understand it, because this is an example that kind of comes with Apis cerana. So this is a honeybee species, that I touched on earlier in the talk with the murder hornets. And this is a species of honeybees found primarily in Asia, and they're smaller honeybees on the whole, kept in smaller boxes, but they still produce honey. They're still a eusocial organism. But they are the honeybee that first evolved with the Varroa mite, right? So this is Varroa jacobsoni, but they evolved with this mite. So researchers thought maybe they had a better way of dealing with the mites, because we didn't see the high rates of death in the Apis cerana colonies that we do within Apis mellifera colonies once the Varroa mite came to our colonies, essentially. So in order to look at what Apis cerana was doing to defend against the Varroa mites, researchers took Varroa mites, they put them within cells of honeybees, and then right before the bees were going to emerge from pupation, right when they should be becoming adult bees, they uncapped the cells to look at what the pupae looked like. And so here we have, these are all the bees that they've uncapped. And so the top row here, these are Apis cerana pupae. So these are the Asian honeybee. And these pupae here are the Apis mellifera pupae. And they've both been parasitized by Varroa mites. But we can see in the top row of the Apis cerana, they're all really dead and really ugly. And they've kind of performed this self-combust mechanism where they're not providing a good feeding source for these Varroa mites anymore. So they've done this almost like altruistic sacrificial behavior, where when they're being fed on by a Varroa mite, they stop growing, they stop producing, so that these mites don't have anything to feed on, and so that they don't continue to proliferate within the colony. Whereas we can see in the Apis mellifera colonies, they're developing like usual. And I just thought that this was really interesting, because we do see within the honeybees that evolved with the mites, this behavior happening, or mechanism happening that creates this defense against the mites that we don't see with Apis mellifera, because Apis mellifera hasn't been around Varroa mites for as long. And so obviously this is something that would take a long time to apply to Apis mellifera, but it is interesting because it's, again, this kind of social immunity, maybe an altruistic behavior that's contributing to the survival of these honeybees in the face of really bad parasites such as the Varroa mite. So yeah, if this could be something that could be applied to Apis mellifera, that would be really cool. But in the meantime, it's really interesting that bees are doing this and that there is, potential for this resistance mechanism. Yeah. Which is right. It is really interesting. So it's almost like they surpass the hygienic behavior altogether and just kind of go to like a shutdown mode. But then the Varroa mites don't have anything to feed on, so they can't keep reproducing essentially.
So are all the mellifera have Varroa mites?
Yeah.
Okay.
Well, yeah, they don't in the picture, but they did during puberty. I think they might have removed them for the picture or whatever. But yeah, I just thought that was a really interesting study, so I threw it in, but it's kind of down a couple rabbit holes and not super applicable, but it's just interesting to see that these different mechanisms within the honeybee colonies could be potentially helpful within colonies that we have at some point. Okay, so moving on from that to the last couple forms of social immunity that we have. The first up is social fever. And so this kind of is the one we already talked about with the murder hornets and the social fever with them. So when the murder hornets infest the colony, you have the honeybees heat balling them and able to heat up to cook the murder hornet so that it can't be a predator to the colony. But we also have this kind of temperature as a defense mechanism also with chalk brood. So when a colony has chalk brood, a paper found that if they have chalk brood, the colony will start to heat up to a temperature, again, to kind of cook out the chalk brood so that the colony is healthier and not having chalk brood anymore. So this is kind of interesting because honeybees do have a pretty high threshold for thermoregulation, so they can perform this heating behavior to try to kill certain, pathogens or parasites. So yeah, this one could be interesting, especially because I think that Varroa mites also kind of have that stress threshold of heat too. So potentially, this could be something used against Varroa mites too, or, it's just interesting to think about all these different mechanisms that they have as a colony to defend against things. And then last up, we have absconding behavior. And this one obviously is the most inducible because it's kind of a split-second decision and it's a pretty permanent one. But as a last-ditch defense mechanism, honeybees can also completely leave a sick colony and abscond. This can happen, in response to a predator in the case of like a honey badger or something like that. But here it would be more likely in response to just a really, really sick colony. So if you have a lot of pests, parasites, pathogens, the colony can just up and leave them behind, which will leave all the, infected wax, infested brood behind, and they'll be able to start anew. But from what I know, this isn't like a really common defense mechanism, which is kind of interesting. But I thought that maybe as beekeepers, we could kind of mimic this response because we can't just let a colony abscond because we want the colonies. But maybe if we removed frames of really infected brood or really, bad frames and replace those with the frames that I talked about earlier, frames that you remove if you have like high honey sources or brood food, you can put them within that colony and potentially just give the colony a new start with healthier wax or something like that. But obviously this one is kind of happens when the colony is really far gone. So hopefully it wouldn't get to this point in the first place. But if you did have a colony that was really, really infested, you could remove some of those frames because obviously whatever is in those frames, Varroa mites or pathogens, are just going to keep proliferating into the colony if you let it. So, yeah.
So freezing frames kill Varroa mites?
Freezing frames does kill Varroa mites. From what I know, freezing frames is good for almost anything except for Falbrood, which, yeah, you've got to burn the frames if you have Falbrood, but fingers crossed on that one. So yeah, so that should be good if you freeze them. And then if you put the frozen frames back within a really healthy colony, they should be able to clean them out fine and the Varroa mites will be dead. So that should be fine. So yeah, overall conclusions about this social immunity technique that bees are using is that, it's really interesting because it's only possible because of their social structure. And so we have these really cool organisms that are just a group of a bunch of different organisms that are really trying hard to keep their colony alive. And obviously so are we. So it's nice to know that we're working together with them, even if sometimes it can feel like we're not. So these techniques are obviously important in colony health. And there are little things that we can do to kind of help them out with this. And I think it's also really interesting just to know, that when people say you need a lot of different floral sources, that there is some science behind that, and it can be because of these antimicrobial properties, not just for nutrition. So there's like a lot of different reasons to be kind of exposing your bees to different floral resources. And as we learn more about these techniques, we can, create healthier honeybee lines and encourage these behaviors to keep the bees healthy. So yeah, that's what I have for the social immunity portion. So I can go in and talk a little bit about my research, which is also kind of social immunity focused, but I can take any questions about like those specific behaviors first if people have anything. Or I can just go straight into the application. Okay. Oh yeah. Yeah, so I think in certain brood foods, there can be antimicrobial properties. I don't know what that pollen would be though. But yeah, I can comb back through that paper and see if they mention anything specifically, but that would be good to know.
It's just like humans, you need a very nutritional diet.
Right.
I mean, one food source, just eating French fries every day, it's not good for you.
There's also someone that just mentioned, there was an article saying that hygienic behavior can actually hinder bees as they chew on infected larvae, therefore acquiring and propagating via prophylaxis. Have you found anything to support or dismiss this?
I haven't. Didn't actually see that article, so that sounds really interesting. I do think there is something to kind of artificial breeding. It seems like that could be, a difficult road to walk in terms of encouraging a behavior, but in a way that it could be like interbreeding. But I think, as far as I know, hygienic behavior is still a pretty positive behavior. So yeah, I can now get into my research. So my focus is a self-removal behavior. So this kind of ties into the social immunity because I have ideas that this could be altruistic, but it's not social immunity in as that it's potentially protecting against pathogens as much, but we'll get into it. So like we touched on, age polytheism in honeybees, this is where the bees perform different jobs at different times in their adult life. And like I mentioned, this is a social immune tactic, but the important thing that I want to draw attention to in my research is this day 21 forager part. So typically, and this is like pretty well researched, well established, bees become foragers around day 21 of their adult life. However, this can be a plastic, thing. So bees can't, so bees can perform different jobs. If, say, you lose a generation of forager bees, these younger bees can actually age up to take on a foraging role if there aren't any foragers within the colony. But in a colony that has, a well-regulated population, the bees will be foraging around day 21. But like I've just said, if you lose a generation of foragers, bees can age up to take on this role, and this is called precocious foraging. And so precocious foraging can happen in response to the loss of a generation of foragers, but it can also happen in response to stressors. So a lot of research has been done on different honeybee stressors, and it's been shown that honeybee stressors just cause precocious foraging as a whole. So if a honeybee is stressed, it's much more likely to leave the colony early. And this can be caused by a bunch of different physiological things, but the important things that I want to draw attention to here are weather patterns and parasites. And so in this, is it like timed? So here in my research, I'm looking at cold stress and hot stress, heat stress, I suppose, and then also Varroa mites. And so in all of these scenarios, these stressors have been shown to cause precocious foraging behavior. And so, how I said it, where bees are aging up to take on this role because there's potentially missing bees, it sounds like a positive thing, right? They're all filling in the jobs they need to. But studies have actually shown that precocious foraging can be really detrimental to colony survival in a scenario where you have these stressed bees aging up to take on this role, these precociously foraging bees are actually just much worse at their jobs. So they forage for less time, they spend less time in the field, they're more likely to die in the field, and they take home fewer resources to the colony. So this is really interesting that this is like not a behavior in the stress bees that's influencing the colony. So studies have shown that this precocious foraging behavior can create a feedback loop that can actually lead to colony collapse. Because you could imagine, this is, you know, kind of a crazy diagram here, but if you have foragers and you lose some of those, right? And so then you have these stress bees or younger bees aging up to take on this foraging role, then you have fewer hive bees able to take care of the uncapped brood, but then you also have these bees that are worse at foraging, And so then they're more likely to die in the field. And so then you're likely to have even younger bees taking on this role. And so then it just keeps going like that until the colony can collapse. So this is that paper modeled it and showed that independent of any stressors, actually, if you just lose a generation, this can lead to colony collapse. So that was really interesting to me. And that kind of plays a role in my research, because in my research, I observed that bees that had been parasitized by Varroa mites were leaving the colony really early, around like day 2 or 3 of their adult lives. And they would just fall to the ground outside because they're so young that they couldn't even fly. Like their wings weren't even developed enough to fly away. So they'd fall to the ground outside and they'd kind of crawl away from the colony until they died. And I thought this was just such an odd behavior because, you know, this can't be very beneficial to the colony. These bees aren't even foraging. They're too young to do even that. And they're just dying outside the colony. And so my thoughts were, because the Varroa mite is parasitizing these bees, maybe this could be a form where these bees are trying to remove themselves from the colony to avoid parasitization. Or it could be, you know, let's see if this video plays. This is a video of the behavior. It didn't play the last time I did this. So we can watch that again. So this is a tagged bee on the entrance of the runway. And so I tagged this bee when she was just emerged. So she's 4 days old.
To her death.
So sad. So she's 4 days old in this, and she does, and it's weird too because you kind of see her back up, and she does do a little wing fly thing. So like maybe she's trying to forage. So I was toying with these different hypotheses of what could be going on, because while it could be, you know, potentially where the bees are exiting the colony because they're infected with mites and they want to get the mites out of the colony, maybe it's something like that. But then I was thinking maybe it is a form of precocious foraging behavior where these bees are just really, really stressed out. And they're stressed to the point where they, you know, the only thing that they can do is try to go forage, but then they can't even do that. So in order to figure this out, I wanted to use different stressors to look at if it's just something that's being driven by Varroa mites and parasites and pathogens, or if it's something driven by kind of stress in general. And if it's stress in general, maybe this is a form of really accelerated precocious foraging, where the bees are just exiting the colony early and dying outside. So yeah, so I used heat stress and cold stress, and then I used Varroa mites. And I used the mites because this is what I had observed the behavior with originally, so it just made sense to kind of keep looking at that. And then I used hot and cold stress because it's easy to work with. And then also, you know, in light of a changing climate, we've got a lot of weird stuff going on, polar vortexes. We don't know what's going to happen with the heat and cold with our bees. So this is my methods, and this we can kind of gloss over because this is really a lot going on here. But basically, I took bees right when they emerged from pupation. So they're baby bees, and I tagged them with individual tags. So I knew the number and color of each individual bee, so I could figure out what's going on with everyone. And so I had mite stress bees and control bees from the same colony. And then I had cold and heat stress bees that I put in an incubator while they were pupating for 24 hours. So I put them in there, and once they emerged, I would tag them so I could see everybody within the colony. And so then my main mechanism of observation for this experiment was that I would stay out at the lab for like all daylight hours, and I would check outside of the colony every hour in this plastic tub that the sides were coated with Vaseline, so they couldn't crawl out if they wanted to, but they could fly out if it's a bee that can fly. And I would check that tub to see if there's any bees that were tagged, and then I would collect them and freeze them for later analysis. So like the image that you just saw, or the video you just saw, that was a video of one of my little tagged bees performing the behavior right into the tub, and then I can pick her up. So I did this for 7 days after the bees' introduction into the colony, because I figured that's about the time that they can't really fly, they're not really doing any foraging behaviors. From what I have read, precocious foraging typically happens from around day 10 or 14 of the bee's life. So that brings me to my results here. So this is kind of a lot, so I'll try to go slowly with it. So essentially I have all of my different stressors, right? I have hot stress, cold stress, and mite stress. And so here we have my controls in yellow or orange, and we have the cold controls here, and then the cold stressed bees. And then we have the mite control bees here that don't have any mites on them. And then we have the mite stressed bees, then we have the heat control bees and the heat stressed bees. And so then on this axis, we have how many of the bees perform the behavior I was looking at, right? The one that I showed you the video of, how many of them are running outside of their colony and dying. And so we can see with every single one of these stressors, the bees are removing themselves from the colony at much higher rates than their control counterparts. So with the cold, with the mite, and especially with the heat stress, we have the bees leaving the colony at higher rates. And so it doesn't seem, based on these data, that the mites are causing a higher proportion of self-removal than any of the other stressors. So this, to me, indicates that this could be a precocious foraging kind of driven behavior where the bees are just really, really stressed out. And so the only thing that they can do is leave the colony. And even though they can't forage, they leave the colony and then they end up dying on the ground outside. So, you know, why does this even matter? Mostly because of the figure that I showed you earlier with the model of the precocious foraging behavior and how that can lead to colony collapse. This could be something that even exponentiates that to a larger degree, right? So if we have these bees that are super stressed out, that are self-removing even earlier within their lifespan, this is creating more gaps within the colony, and it's creating this behavior where you just lose a lot of bees. You have, and then this can also contribute to precocious foraging behavior and more of these precocious behaviors. So it was posited in the original paper that showed that this can lead to colony collapse, that this could be kind of a form of colony collapse disorder, because in this scenario, you're losing all of the adult bees, right? They're all leaving the colony to try to forage, and then even the younger ones are trying to forage, and the bees are self-removing. They get super stressed out because they can't do anything within the colony. And then you're left with the queen and a few of the adult bees that aren't really, really sick and have to leave the colony, right? So, you know, this is some interesting data that I have. And so hopefully as I move forward within my program, I can continue to back up the idea that this is a precociously foraging driven behavior. And then ultimately what I would like to do is kind of model this behavior to see if it can lead to colony collapse and at what stress thresholds we can expect this colony collapse to happen. So if, you know, we can keep our colonies under that stress threshold, whatever it may be. So if it's a certain number of mites, a certain pesticide content, whatever it is, if we can keep our colonies under that threshold, then maybe we won't be seeing this behavior that ends up being really negative towards the colony. And so, yeah, then recently, actually like this past week, I just finished a trial in which I did pesticide stress. So I used amitraz and I used chlorothalonil, which are different pesticides found within honeybee colonies. So I'm really excited about that too, because that's also seems to me to be like a pretty applicable stressor, because all of us are encountering that within our hives. So hopefully that'll help us continue to understand what's happening with this behavior and how it could be influencing honeybee survival and colony health. So yeah, with that, I'd like to say thank you. And I can take any questions.
Any questions online?
How do you think this relates to packaged bees?
Ooh, I have no idea.
It's just a bunch of bees out of the
Right.
Yeah.
You don't know what ages they are.
Like here, make a hive.
Yeah, that is a good question. I would think that if, you know, in an ideal world where they're not really stressed out packaged bees, if you have them and they're pretty healthy to start with, they can probably fill in all those different jobs that they need to fill in to hopefully start the colony off strong. And then as they start producing, you know, more bees, it can continue to even out that way. But if, you know, if you have like high mite concentrations in your apiary or anything like that, you could start to experience stress within that colony faster. And presumably, yeah, you could lose a colony faster if you put a package in a stressed place, which that's an interesting thought. I hadn't thought about that.
How did the storm affect bees long term? Ice storm,
Yeah, that's a good question. So I didn't actually do any like observations long term of that, but based on what I think we had, we have about 60 colonies out at our apiary, and I think we lost 4 or 5, so it wasn't too bad. But over time, we did kind of see like more losses within the summer. So you could almost think that if this is, that cold stress
Stressor.
that you're getting, it could kind of start to spiral and lead to that. So maybe in a place where bees aren't ready for that kind of cold stress, that could be an expectation. Yeah. Yeah.
You mentioned earlier that freezing the brood would get rid of Varroa mites,
Uh-huh.
and it helps everything except Falbrood.
Yeah.
So I have 2 questions. Does the brood die if you freeze them? And what is Falbrood?
Yeah, so the brood does die if you freeze it. So that's kind of a sacrifice that you're making, right? You don't want to freeze the brood unless you have really high mite concentrations so that you're getting rid of as many mites as possible. And then Falbrood is a bacterial infection. There's American and European, and one is worse than the other. I think American is worse. And so they're bacterial infections that get in the brood, and they make the brood really like stringy and sticky, and they kill it. And then because it's bacterial, it can be spread really fast throughout the colony. And once it's spread, it infects basically all of the materials that have touched it. And you can't freeze it away. So you kind of just have to burn the frames, particularly with the American Falbrood. European, really strong colonies can deal with European Falbrood fine. And normally is a problem when you have weak colonies, I think.
Questions?
We do have an online question asking, where can we follow your progress, results, and publications?
Wow.
Yes.
Thanks. That is a lot of pressure. Yeah, so I don't have anything out yet, but I stopped sharing. But I have an email that I can give out if people are interested in that. And then, yeah, hopefully I can have more updates for you as time goes on. I know that with the Rangel lab, I think we're letting out little updates of each person in the lab through like the Texas Beekeeping Journal. So I think I can write an update for that next year. So maybe we can check in via that, and then other beekeeping meetings and such. But I don't have any sort of social medias yet.
Do you graduate next year, or when do you graduate?
No,
In theory. In
So I'm
theory.
just starting my 3rd year. And so probably within the next 3 years, I can hope to kind of get everything.
We can
wrapped up.
check back in like
Yes,
in a year.
yeah, we can be friends. Yeah. That's a great question. Yeah, so pesticides can be found in honeybees in a lot of different ways. And one of the ways is within the wax of the honeybee colony, because bees will be in contact with pesticides in the field, or you'll put, amatraz strips right in the colony and it'll kind of infuse with the wax. So the way I did it is that I had like brand new frames with the wax foundations, and I kind of brushed it with wax, and then I would spray it with a pesticide concentration, and I used a field relevant concentration, so what you would typically find within a honeybee colony, but the high end of that, so that it could be like pretty stressful, but nothing that you wouldn't see within a honeybee colony. And then I sprayed it and I kind of painted it again with the wax, so it was just one application, and then I put it in the hives so the bees could build out the frames, and then they would rear the brood within that pesticide wax. So they'd be in contact with it in a way that they would normally be in the field, to see kind of how that affected their behavior.
Fascinating.
We have another question from the online audience. Stan Gore wants to know, how long have you been a beekeeper?
That's a great question. So I got into beekeeping and bee research when I was in undergrad, I think my sophomore year. So that would just mean what? Going on 6 years now, so not too long. But I was in Tom Seeley's lab as an undergrad, so I had a good teacher. Awesome.
All right. Good job.
Cool. Thank you.
All right. Thank you so much. I got the opportunity to hear Jordan at the bee school the other week, and I just thought it was so fascinating. I was just kind of geeking out with all of the information. So that should be great for us today too. All right. Well, so next up, I'm going to invite Joe Powers up. He is going to give you actually an overview and share with you our upcoming mentoring program, as well as, if you hang out long enough, he's got all the prizes. So you have to be present to win. Don't leave yet.
Oh, do you want mine?
Yeah. Here's a mic,
That's true.
so you can talk about mentoring.
Feel scared.
Put this on your collar.
There, okay. Can you all hear me? Okay, good. All right, so let's do the mentor program first. So, oh, I didn't bring my paper up. Hold on. Oh, there it is, right there. So we're starting a mentor program in Houston Beekeepers, which we really haven't had formally before. Or they probably did. We've been around since 1959, so I'm sure at different points in our history, we've had a mentor program, but currently there's not one active. So we're starting one up. It really and truly starts at our November meeting. At the November meeting, we're endeavor between now and the November meeting to get everybody to go online and get the forms that Kyle's going to put on there for me. One is a form that you fill out if you want to be a mentee. The other is a form you fill out if you want to be a mentor. And the qualifications to be a mentor is you've got to have kept bees a hive for like 1 year. You don't have to be a 40-year beekeeper. You just have to have them for a year. And it doesn't have to be like this last year. Let's say, for example, you're one of the guys in the audience and you had bees several years ago and you're going to restart doing bees. You already know how to do it. You can still mentor somebody else, even if you're just starting over, you know, because a lot of us have done that, had bees and then had a problem, skip the bees for a while and then come back to try again and got better as you do it. So there's a description of the mentor program. That's, I think, is it on our website somewhere yet? It'll be on our website. Coming soon. And there's a hard copies back in the back. So you guys can get a hard copy of this and read about it. It's pretty straightforward. And the thing is, the way we want this to work is you're a mentee for 1 year, learning how to do bees. At the end of the year, you're ready to be a mentor and teach the next guys how to do it, right? So you might not be quite comfortable doing that, but I guarantee you when you're trying to explain to somebody else what it is you're doing, you actually have to think about it a lot harder and you learn as you go while you're trying to teach somebody else what you're doing. I'm sure Jordan has experienced that trying to explain to us social stuff here, you know? So, okay, door prizes. Must be present when we have 4 door prizes. Cool hat, Houston Beekeeper shirt, field evaluation journal, or a hive tool that's actually pretty darn sharp. So be careful as you win that one. Okay, random number generator. First number was 8, black shirt. You won. Come take your pick. All right, perfect. Oh, you weren't that way. Sorry. Next number 3. That's you.
Sure, please.
Okay, it's a large, so should be good. All right, next number 9. So cool hat or a field evaluation journal. This is actually kind of handy if you're just starting off on bees. Probably. All right. All right, and then the last one. No, can't be the same person. Number 1.
Oh, there you go. Oh, I get it.
And you need the hat, just like me. Okay. Do you want to tell them about the November meeting too?
Yes.
It's going to be here
Yes.
and next year.
And next year. Okay. One more.
Oh, Mike.
It's all right. I got one. All right. All right. So last things of note, I think on our last slide, our next meeting is going to be here and online. So we'll be back here at the Young Center. It is going to be a fun potluck. So we're going to kind of organize who brings what. We'll kind of provide the basics, but we're looking for all kinds of goodies. And of course, if it's honey related, we'll really like it. We'll also be able to share with you then the nominations for our new board members and really just kind of kick off the new year. We'll go back over the mentoring program and hopefully connect up a couple of people in person to their new mentors. So we're really excited about that. Next year, we will be back at Bayland. So for those of you who were members, we were meeting down at the Bayland Community Center. It's kind of close to Bel Air, right? Yeah, just west. We will be back there next year. We've paid our dues and they're ready to have us back. And so we'll be in person. We will also try and continue our Zooming for those of us that can't come in person. But we're gonna, try and see how that works out for next year. So we're really excited for it. Anything else that I need to mention? Oh yeah. If you joined late, Texas Beekeepers Association is having their big conference, which is November 6th and 7th. Thank you. Definitely try to attend. We don't always get to have it as close as it is in Galveston. Sometimes it ends up in other places in Texas, and it's a big state. But it should be so much fun. Lots of speakers from all over the United States. You can shop, you can spend time with other beekeepers, and a bunch of us will be there too speaking as well. So we hope to see you there.
Go to it and you see a speaker that you really like.
Oh yeah.
Tell somebody on the board about it so that we can try to come talk to people. That's how we.
Exactly. So that's how we found Jordan. So when you go, if there's ones that you really think were just amazing speakers, we will be there, but we can't be in every class. So please let us know if there's any in particular that you see that you just think, you know what, we have to get this person to come speak at Houston Beekeepers Association. Because you'd be surprised how many people we can get to come join us virtually as a speaker, or sometimes drive in and be in person. Look out. So it's really a lot of fun, especially this last year. We've had so many speakers that we probably never could have gotten if it wasn't virtual. So it never hurts to ask. And in fact, very rarely do people say no. So we're just really enthused for the next year. So with that, I think that's all of our announcements for today. Kyle, anything else? Okay, we will post this up so we'll have it recorded, and have an amazing month, and we'll see you back here in November. Good job.
Thank you so much.